Hello friends, this video on molecular basis of inheritance part 17 is brought to you by examfear.com. No more fear from exam. So now we are going to talk about the next important process, that is transcription. So till now we saw that how DNA creates a copy of itself. So now we have two copies of DNA. But DNA directly cannot synthesize proteins, but our aim is to synthesize proteins because proteins directly impact the traits which are seen in an individual. So if DNA cannot directly synthesize proteins, so what can be done? So RNA needs to be synthesized from DNA. So that is what we are going to look at in transcription, that how do we get RNA out of DNA. So what is transcription? It is a process by which a particular segment of DNA is copied into RNA for protein production. And why does this happen? Because DNA directly cannot synthesize proteins. So it has to generate RNA, and this RNA can then directly code for protein synthesis. So transcription is all about how do we get RNA from DNA. So as we discussed before also, that DNA is the storage of genetic material. So purpose of DNA is to store the genetic information because it is chemically, structurally more stable. But in order to control the traits with this genetic information, if somebody wants that the eye color of a person should be similar to what it was for his father or mother, so that means we want the genetic information to take control of the traits. So if we want that to happen, then the information which is stored in DNA needs to be expressed. And for expressing that information, proteins need to be synthesized accordingly. And for that protein synthesis, RNA has to be created, and that RNA will code for protein synthesis. So transcription will actually tell you how we can get a copy of RNA from a particular segment of DNA. It is not that the entire DNA will get copied into a RNA. A small portion of DNA will give rise, or it will result in the formation of a segment of RNA. So this is the first step of gene expression. So whenever we talk about gene expression, that is expressing the genetic information, RNA comes into picture. So the lead role is now taken over by RNA when we talk about gene expression. Now when we are talking about this synthesis of RNA from DNA, the discussion would be incomplete without talking about the central dogma of molecular biology. So let us see what was the central dogma of molecular biology. It was a hypothesis which was proposed by Francis Crick. Yes, Crick is again the same scientist who was involved in finding out the double helix structure of DNA, who was also involved in giving the hypothesis that the mode of DNA replication is semi-conservative in nature. So he again gave this hypothesis of central dogma. So according to this hypothesis, it explains the genetic information flow in a biological system. As per this, in any biological system, the genetic information will flow in the following manner. And what was that? As per this, the genetic information will be stored in DNA, and then it will be expressed in the form of RNA, and then this RNA will synthesize proteins as per that information. And these proteins in turn will control the traits which are seen in an individual. So this was the central dogma of molecular biology. So as per this, three major classes of biopolymers are involved in this entire process of genetic information flow. And what were the classes of biopolymers? Nucleic acids, that is DNA and RNA, and proteins. So as I said, the three biomolecules or biopolymers which are involved in this case are DNA, RNA, and proteins. So this was what was proposed by the central dogma. So as per this, since we had three classes of molecules, so all of them could be converted, or they could be transferred from one to another. So a total of 9 transfers were possible as per simple permutation and combination. Since we have 3 of them, so DNA can be transferred to RNA, DNA can be converted to protein, RNA can be converted to protein. So that means there could be total 9 transfers which can be possible, out of which 3 general transfers which take place in most of the organisms. Because here we are not only talking about human beings, we are talking about all biological system, any biological system, whether it is complex organisms like human beings or simple organisms like bacteria or any other form of life. So this is universal for all biological systems. So three general transfers which take place are: first is DNA to DNA. That means, what does that mean? DNA to DNA is nothing but replication, which we spoke about just now. So this is the process of replication. Next is DNA to RNA. So that is what we are going to talk about now, that is transcription. And the third one is RNA to protein. So that is known as translation. So this process is called translation. This also we will discuss after transcription. So these are the three general transfers which take place in most of the organisms. So wherever DNA acts as the genetic material, these are the three steps which take place for the flow of genetic information. However, there are some special cases which have some special transfers happening there. For example, some viruses where the genetic material is not DNA. So if the genetic material is not DNA, so in that case there won't be replication of DNA. So this flow will not happen in that case. So what would be the flow in that case for special transfers? So for special transfer, RNA is the genetic material. So RNA replication will take place. So RNA will create a copy of itself, and then RNA will create DNA. So it is just going to be the opposite of transcription, and this is known as reverse transcription. It is called reverse transcription. But please remember, it doesn't happen in all the cases. It happens only in special cases like viruses. And the third step would be DNA to protein. As I always say that DNA directly cannot code for proteins, but viruses are an exception where RNA acts as the genetic material. So it happens only in special cases like viruses. So these are the three special transfers. Now, the remaining three transfers which I was talking about, they are the unknown transfers. That means whether they are possible or not, that is not yet known. So basically these kind of transfers have not yet been known to take place anywhere. And what are these transfers? That is protein to DNA, that means conversion of protein to DNA, a complete the reverse process, or the conversion of protein to RNA, or the conversion of protein to protein, that is creating copies of the proteins. So all these transfers have not been known yet. So these are the unknown transfers. But the general transfers take place in most of the organisms, and special transfers only in some special cases like viruses under special conditions. So these are the various ways the flow of genetic information can take place in any biological system. However, what we will deal here is the general transfers. So we will talk about only the general transfers, where DNA replication we have already covered. So this process was DNA replication. DNA to RNA, this process is transcription. And RNA to protein, this process is translation. So these are the three processes which we will discuss in this lesson. So replication we have already covered, transcription we are in progress, and translation we will study after we are done with transcription. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.